So you guys ready? Ready? All right. Come on in, everybody. A room full of my favorite data nerds. All right. My name is Stanley Blum, and I'm chairing this session, but also speaking first. This is Occurrences are Neither Specimens or Samples, Data Modeling Challenges and Opportunities for Information Storage and Exchange. And so nicknamed Occurrences, thing or not a thing. And I'm just going to get right to it, I guess. Yeah, it's not advancing. There we go. All right. When we created the Darwin Core way back in the early 2000s, we had two driving purposes and three major classes of biodiversity, primary biodiversity information. The purposes were to expose as much data as possible relevant to documenting species distributions. And then the other one was to enable researchers to find the stuff that they needed to study, so the discovery part of metadata. And so those are the two things up at the top. Down at the bottom, we had sort of three major categories of information: observations, specimens with natural geolocations, natural occurrences that would be relevant to species distributions, and then specimens that had unnatural locations that still might be useful for people that need to study certain kinds of materials. So a few, oops, gotta do that. Oh, come on. There we go. A few years later, we went from what we had been doing before the Darwin Core was doing a lot of normalized database design targeted for designing collection databases. But when the web really started to happen, information integration really became possible. Distributed queries or harvesting was the thing, and ultimately Tadwig decided to go into the semantic web as the primary way of documenting concepts and semantics for our community. So after a lot of discussion online, Steve Baskoff and Campbell Webb came up with this Darwin SW, the semantic web version of Darwin Core. And come on. So this is sort of my entity relationship version of that. And let's see, is this working? Yes. All right, so the occurrence was explicitly recognized as a thing that represented the intersection between an organism and a collecting event or a sampling event, and the result was either an information artifact or a material sample. So these were, in essence, subtypes of this token is evidence of that organism in a place and time. Let's see. All right, so then going back to sort of how we were evolving things and where we were coming from, I guess, in the evolution of collection management databases. The first ones were super simple: flat table for your specimens, maybe a taxonomic controlled vocabulary. But then, of course, if you needed to track the parts of your specimens, let's say you're dealing with higher vertebrates, say mammals, typical preservation might be skin, skull, and a frozen tissue collection, a frozen tissue sample, and generalize that to one-to-many preparations as part of the specimen. This is not, there we go. So this in a table form looks like this, where all the parts are pointing to their material sample, which actually is a virtual thing at this point because it doesn't exist anymore. All right, so actually the first kinds of things that were done as we were normalizing specimen databases was the separation of specimen and locality, where there were disciplines where multiple specimens were produced from a given locality. But different disciplines treated the time component differently. In ichthyology, the saying was just like the Zen thing, you can never step in the same river twice. So the time was put into locality. But if you're a paleontologist, you go back to the same place season after season after season, so the time went with the specimen. And to reconcile those two, we came up with this collecting event. So we ended up with this sort of a cascade of one-to-many relationships, locality, collecting event, specimen. And if you put that out on a table simply, you would have something like this, and notice that you have these IDs. We're obsessed with IDs, of course. And these are what links these three tables together. But normalization is great for discipline and consistency and efficiency in local data management, but it's not necessarily the concern of information integration, because you're basically pulling together, you're optimizing for queries, you're optimizing for basically responding flexibly and quickly. So when you denormalize that and flatten it for publishing, basically those IDs in those middle things can go away. So would the same thing happen to occurrence if we basically are publishing occurrence? That's a sort of a rhetorical question at this point. All right, so I titled my talk basically trying to have how will use cases guide our modeling. So these are just some of the typical use cases that I've experienced over time. Let's say the basic simple one, a mouse is collected, tissue sample taken, whole specimen is prepared into the typical skin and skull, one collecting event, one organism, one occurrence, 3 material samples. Then we get a series of the kind of one-to-many, where many specimens are produced in collecting events like a malaise trap, or in botany, where people might take 3 cuttings from a plant and distribute them to 3 different institutions, and those identifiers that were on all of that, those collecting events need to be maintained as they tie them together as they get cataloged later. The thing that kind of really, in the Darwin SW, the semantic web, the use case that was really driving the change away from normal collection databases were the observation kind of things, or the collections of the same organism repeatedly over time. So Steve was talking about, there's a famous tree on Vanderbilt campus that you can go and photograph at different times of year to see the inflorescences happening. So you could collect them. So you could collect leaf buds, leaves, flowers, fruits, all at different times of year. So single organism, multiple occurrences, multiple collecting events, multiple material samples. Same kind of thing could happen with an organism that's tracked over time, like if you put a biologger on a turtle or a tuna or a great white shark or whatever, and track its locations over time. So in that situation, we ended up with a very different kind of a relationship where the organism could be encountered multiple times. It wasn't like in many of the collection databases where the organism is basically encountered in a terminal event. All right, and then finally, here's the last use case that really kind of changes things quite a bit, and it's the environmental DNA use case, where you can take multiple samples in a collecting event, and each sample is a multi-species sample subjected to some sort of sequencing. the sequence is blasted against reference database of sequences, and you can end up with inferences of many kinds of organisms out of that single material sample. So, so we had a task group that was sort of stepped up to try and model out all the different classification, a consensus classification of material sample. And this is not the consensus that was arrived at, because we didn't. It was too difficult with people coming from various different perspectives. This is just one that I liked. But it's, like I said, it's very difficult to arrive at that. And so my little takeaway for this session is that I think our highest level classifications and how you present data to an end user who's taking from an aggregator is very purpose driven. What is their purpose? What are they trying to do with their data? And then at the other end, down at the bottom, what we should be doing is looking at minimally classified, well-known communities of practice. How do they communicate with each other, and what are their sort of standard methods and material samples that are most familiar to them? And it's in between there that we'll end up with some reconciliation of this. And with that, I will end, and I've got a couple minutes for questions. Lights?
Can I start still?
Yes.
This is very cool, and the last slide was very nice to see. And I was thinking about this a lot, and you capture it very well. So there is sort of use case and context oriented databasing, and then there is community of practice databasing. But I was thinking maybe there is a more neutral kind of standpoint that is more appropriate for TADWIG and data aggregators like GBIF. Is that could we maybe just not have perspectives and capture it neutrally from the point of view of documenting evidence? And I try to look outside the TADWIG box and biodiversity data box, and two contexts of where this is very important is crime evidence and medical diagnostic symptoms. If you think about kind of biodiversity as organism or quasi-organism, and you document health, you don't have certain sickness in mind, you just document symptoms neutrally. What's the temperature? Is there a rash or not? And if it's a crime scene, if there is a dead body, like specimen, that's one type of evidence. If there is only a track, then it's another type, and we have to weigh them differently. And I was thinking, is there any room for this kind of thinking in your schema, like the last slide you show, or is it completely like un-Tadwigian to think?
No, I think that's very much towards the end that we might be working for as we capture and manage data. The question is then, how do you represent it in an aggregator to be as flexible as possible? And that's where I guess the GBIF data model is going. So with that, I've run out of time. And next up is a recorded presentation from Christian Bölling. Can you start that, the recording?
Darwin Core Standard defines occurrences as an existence of an organism at a particular place at a particular time. Setting aside the fact that the term organism has a technical definition in Darwin Core, the concept of plants and animals, biological individuals in general, occurring in particular places at particular times is part of our ordinary, everyday experience of the world around us. Various methods of inquiry are employed to record such occurrences, ranging from unaided visual observation to sophisticated methods of environmental DNA analysis. In this example of an occurrence dataset, contributed via OBIS CMAP to the Global Biodiversity Information Facility, GBIF, time-stamped data on the location of individual Arctic terns along their oceanic migration routes was collected by fitting individual birds with miniature geolocator devices weighing only 1.4 grams. There is plenty of evidence that information on the occurrence of biological species in space and time is essential for many fields of scientific research and for many applied aspects of understanding and managing natural resources. The annual science review conducted by GBIF gives, along with other documentation of GBIF data use, a good indication of the diverse fields of application that occurrence data serve. Like, for example, in this case, where current ecological niches of species are modeled based on occurrence data mediated through GBIF. However, documentary evidence suggests that there are a number of pitfalls and problems when working with occurrence data. A couple of pointers to those discussions are shown on this slide. So, on one side, the concept of occurrences seems entirely natural, intuitive, and useful. On the other side, the ongoing discussions among biodiversity data publishers and users of the data call into question if the documented conceptual basis for occurrences is adequate and if occurrences, as an element of biodiversity data exchange and computational knowledge engineering, are sufficiently understood and standardized. Many of the discussions touch upon conceptual questions regarding the nature and properties of occurrences. Among those questions regarding the ontological status of occurrences, which have been characterized as artificial or intangible, and how occurrence instances can be delineated, given the common practice to aggregate data on occurrences for groups of individuals and along spatial and temporal units. Other questions concern the nature of the relations between occurrences and the means employed to observe them and the artifacts produced in the process. In this contribution, an analysis of occurrences is proposed that offers a connected, ontologically grounded view on these issues. Imagine we had the ability to track the location of a biological individual over time with perfect precision. Then we would be able to observe that the biological individual occupies, from the start of its ontology to the end of its ontogeny, a certain region of space. For a fish, this would be a fish-shaped region of space. For a tree, that would be a tree-shaped region, and for an ant, that would be an ant-shaped region. Through time, the location of the biological individual, in terms of the region of space it occupies, continually changes. The diagram shown here is a simplified conceptual illustration of this view. At each point in time, a biological individual occupies a certain three-dimensional portion of space, a volume, symbolized here as a one-dimensional region of space variable. That region changes continuously through time as the individual moves about. Based on this view, the ontogenetic occurrence of a biological individual can be defined as the spatiotemporal region occupied by the biological individual during its ontogeny. Spatiotemporal region is to be understood as the trajectory of a three-dimensional spatial region as it changes through time. At any point in time, the region of space that is occupied by the biological individual can in theory be specified with respect to a given geographical coordinate reference system, like for example the World Geodetic System WGS 84. Likewise, corresponding time points can in theory be specified with respect to a given temporal reference frame, like for example UTC. Any given segment of this trajectory constitutes a part of the overall spatiotemporal region occupied by the organism during its ontogeny. Such a segment is characterized by a lower and upper temporal boundary and the corresponding trajectory of the spatial region that is occupied by the organism within that time interval. Given the definition of an occurrence in Darwin Core as an existence of an organism at a particular place at a particular time, an instance of an occurrence as defined in Darwin Core could be understood as a certain segment of an ontogenetic occurrence and vice versa. That is, any segment of an ontogenetic occurrence constitutes an instance of a Darwin Core occurrence. In the following, the term occurrence is used according to this interpretation. This is to say that occurrence refers to a specific trajectory of a spatial region in a given time interval that is a specific segment of an ontogenetic occurrence. This interpretation extrapolates to groups of biological individuals. For a group of individuals, the spatial region would be aggregated by the individual regions of space occupied by the individuals in the group. Likewise, so-called species occurrences can be subsumed as the occurrence of at least one individual that has no identification. From a practical point of view, the precise spatial region that a biological individual occupies at any given time may be difficult or impossible to determine, but this does not stand in the way of the definition of occurrences as segments of the spatio-temporal region occupied by an individual. The precision to which that spatial region can be determined depends on the methodology used for observation and the available raw data. This means that usually we are only able to assert that the biological individual occupied a spatial region that lies within a spatial region that is specified, or a projection of that region. For many use cases of occurrence data, this is of no consequence, as they require a specification of the spatial region only at lower levels of resolution, including projections of a spatial region onto a two-dimensional geographic frame of reference, or abstractions like point coordinates or polygons. There are similar considerations regarding the specification of temporal regions for an occurrence. For temporal regions too, the precision with which they can be determined depends on the methodology of observation and the available raw data. Often, the temporal region of the occurrence can only be approximated by a containing temporal region, but especially with machine-based detection techniques, the temporal region that is specified may, on the contrary, actually only indicate a point in time at which an incoming signal was recorded, for example, a GPS signal, a photograph, or a sound recording. Let's see how the first two examples given for the occurrence term in the Darwin Core Standard would be interpreted against the backdrop of this model. The first example of an occurrence is given as a wolf pack on the shore of Turan Lake in 1988. In terms of spatio-temporal regions, this would get interpreted as the assertion that an unspecified number of wolf individuals, which in fact belong to a larger social structure as a pack, occupied a combined spatial region contained within the spatial region occupied by the locality identified as Turan Lake for some interval of time contained in the time interval indicated by the year 1988. In the second example, a virus in a plant leaf, the reading is similar. A virus occupied a spatial region bounded by the spatial region occupied by a plant leaf, which in turn is bounded by the spatial region occupied by the New York Botanical Garden during some time interval bounded by the 29th minute of the 15th hour of October 23rd of the year 2014. What this transformation of the expressions exposes is that the underlying semantic structure of these and similar commonplace statements about an occurrence can be expressed in a uniform way when invoking the spatio-temporal regions occupied by the biological individuals at the bottom of these observations. An important consequence of conceptualizing occurrences as segments of the ontogenetic occurrence of an individual is that these entities can be arbitrarily segmented or aggregated by segmenting or aggregating the corresponding spatiotemporal regions. The result of any such operation would be valid instances of an occurrence. An analogy would be the operation of forming or combining subsets of a given set. Any such operation will yield, again, a subset, and there are no wrong or right subsets unless additional criteria are employed. Let's expose some examples that follow from this. Observations of a tree that are 1 minute apart would count as different occurrence instances. Observations of a snail 1 meter apart would count as different occurrences of this snail. The changing positions of each fish within a shoal of fish are occurrences in their own right. Likewise, the occurrence can be specified on the level of the shoal with its spatiotemporal region corresponding to the aggregation of the spatial regions occupied by each fish in the shoal. Note that these examples are intended to showcase the consistency of the model. Knowing that trees don't move, recording a tree's position every minute is usually not informative. What these examples highlight, though, is that the model provides a uniform basis to split or aggregate occurrence data. Regarding the identity of occurrences, the model has a clear reading. Occurrences are identical if and only if they correspond to the same spatiotemporal region occupied by the same individual or individuals. Consequently, approximations or abstractions of the spatial and temporal coordinates, which in many cases reflect inevitable methodological limitations, may present challenges for distinguishing occurrences when data is integrated from different sources. The fact that the observed individuals in many cases are anonymous, that is, that they cannot be re-identified, means that identity of occurrences can usually not be established, and additional knowledge is needed to count distinct occurrences or individuals. However, at the same time, this criterion for identity means that if observations are sparse, which they are in most cases, one can infer that the occurrences that are being observed are distinct. In this contribution, an ontologically grounded view of occurrences is proposed, and the consequences of the proposed model are explored. According to the model presented, occurrences would be accepted as real entities, unfolding in time and ontologically dependent on the biological individual, which persists through time. Identity of occurrences can be defined in terms of the identity of the spatiotemporal regions and of the participating organisms. Occurrences, according to this model, can be segmented or aggregated in terms of the spatiotemporal regions they represent in arbitrary ways. The result of such an operation would be related but distinct occurrences. Data about occurrences can and will usually only be approximate, which reflects both methodological limitations as well as pragmatic considerations regarding the use cases for the desired data. Therefore, information about the kind of approximation, about the observation methodology, and contextual biological knowledge are instrumental to analyze and integrate occurrence data. In many cases, the observed biological individuals remain anonymous, which leads to raw data that is routinely aggregated for groups of individuals. This may present challenges when additional data about individuals is to be shared. An occurrence concept anchored on the level of biological individual, with a clear way to aggregate data for groups of individuals, may be helpful for indicating ways to handle such data consistently. As a next step, it would be interesting to see if the occurrence concept introduced here is compatible with how occurrence data is currently structured, and if the model can be used to identify possible improvements to those structures for more reliable and consistent handling of occurrence data and computational approaches to biodiversity knowledge engineering. Thank you very much for your attention.
And with that, I think we are right on time for the next presentation from Kathryn Hall and co-authors And I believe we're also playing another video. Is that correct? Do you have a video up next?
I can talk live, Stan.
Oh, there she is on Zoom.
Hello.
Turn
Greetings.
it up.
Can you hear me?
Yes, we can. Can you hear
All right,
us?
I can hear you. I'm going to turn my video off because you don't need to see me in my lounge room. Although I've got a screen share, so I don't think you need to see me, but that's all right. So share. Can you see?
Yes, we can.
And you can
You're
hear?
on. You're on. Yes, we can hear. All good.
Oh, good morning. Hello, everyone. G'day from Australia. I'm sorry I couldn't be there, but I'm absolutely delighted to be part of this symposium, and thank you for accepting our talk. This is a topic that has been quite a discussion for us as part of the Australian Reference Genome Atlas project. And I'm really happy to present to you today the interpretation that we've taken around occurrences and show you how that manifests in the application that we're building, which is an index of genomic data which is relevant to Australia's purposes. So the Australian Reference Genome Atlas project has been conducted in partnership between the Atlas of Living Australia, the Australian Biocommons and Bioplatforms Australia. We have also received funding from the Australian Research Data Commons, and we are what is known as an NCRIS facility, which is a facility which is available to support researchers and their research in Australia, and that's funded by the federal government. So the big question for the day is, what is an occurrence? And today I'm going to talk about what we in ARGA are interpreting as an occurrence. It was really great to hear the introduction from Stan and also to hear what Christian was saying, because I think we're thinking very similarly, but maybe slightly differently, in that I heard that Christian was talking about occurrences being entities, but in ARGA we've tried to disambiguate the entity itself from the occurrence. So we'll go back to this definition again, which we've already seen, what a Darwin core occurrence is. But I'm just going to flick back one step to the Oxford English Dictionary, because I always find it useful to make sure that I understand what a word means. And so I turn to my dictionary. And in the dictionary, we can see that, and I particularly like definitions 2 and 3, but it's the fact of something occurring, which it sounds very simple, but if we have a look at that, there's some things in common with the Darwin core occurrence definition, but that are very important that we need to draw out. And they are that it is the fact of a meeting or an occurring or an encounter. So it's us as agents encountering something that forms an occurrence, and it is a single time-bounded event. And that is in common with what Christian was talking about. So what's the relevance of that, and why do I care about it's this occurrence or this meeting of things? Well, the reason is that we're interpreting something which I'm nominally calling occasions. So an organism, as we know, is there in time and space, and it's there whether we know about it or not. But when we come to know it, that's our event at which we meet it. So it may be there, but we have to know about it. And we may meet it, but it's not really useful to us for our analysis purpose unless we've documented that. And we're interpreting that to be the occurrence. So we've got metadata around that. In our model, we're saying that the first time we meet it is event 1, and then the next time is event 2 and 3, and so on and so forth. So in ARGA, what we've done is we've interpreted occasions, which are the event of meeting an organism, and then the information around it, which is the occurrence block. So I'm going to present to you a little bit about the data model that we're using, and what we've tried to achieve with it is to just respect that hierarchical and serialized nature of the times in which you may meet an organism throughout your research journey. And we've tried to use the Darwin core formatting, and we're also importantly wanting to have a consistent model that we can apply across a number of different data sources that we're ingesting from different repositories. And so we're using an event-based structure to do this. What I was trying to draw out in the beginning was that we're taking the occurrence as being different from the actual organism itself. And so we had to model the material entities, the actual organism, and all the derivatives from it. And so you can have a material sample, which you may take from your organism, and then it becomes a specimen when it goes into a formal collection. You may subsample it, take DNA, amplify that, and then eventually you'll get data. So there's a whole number of derivatives that you can make from any given organism. And we saw earlier some of the other ways in which you might meet an organism. But for us, we're looking at the first event where we begin to know about our organisms is at the point of collection. And that's when the material sample is taken. And in that event, or the fact of collecting it, is something that's fixed in space and time, which is that time-bounded part of the definition of occurrence. Then, as I said, it goes on to become part of a formal collection in an accession event. So we've defined a number of standard events that may happen within the trajectory from going from collection through to creating genomic data. They're a physical collection, accession into a formal registered collection, subsampling, DNA extraction, amplifying, sequencing, assembling those sequence data, annotating them, putting them into a repository, and then possibly even downstream reuse where they've turned up in another publication or been reanalyzed. So the way this turns out or manifests in reality is that we take this very simple example where we've got a single specimen that we just got something like, say, a CO1 sequence or an ITS2 sequence from, so single locus data. You've got your organism, you took a sample in the field, the first material sample, which would have an identifier, moved into a collection where it got a registration number, subsampled, there'll be lab tracking numbers, and you can see all the different events. Then mapping on, finally, we get an alignment file that can be reused in some other publication. That looked really simple. It can also be extended out then in a very complex scenario where you've got a whole genome being made. You may end up creating a lot of DNA libraries, may do a lot of subsampling of your initial specimen, but it doesn't really matter. The same steps can be applied. This is very tiny, so we won't worry about it. This one's even tinier. But the main thing I want to talk about here is how we have used the two Darwin core blocks together to show that there was an organism which continues to persist in time and space, but there are these points in which we encounter or meet. So these occasions that we're modeling. So to look at this in more detail and show you what we've actually done, you have your event where you collect it, and you have an extended occurrence block which you can build where you can collect all of that metadata around it. And this is time one. This becomes an event and it's linked up via its material sample ID. And then the next time you see it is when it's in a collection, and you can link these two events, and you can also create a new extended occurrence block. In that way, you're collecting different information about it. So this may have been collected on the Great Barrier Reef, and then this has been deposited into Queensland Museum collection. So you've got different identifications, different locations, different agents acting on it, and all sorts of, so you can actually tailor the information to what is actually occurring in that block. To continue along, obviously, we go through all of those stages along the timeline, and you end up in the final stage here at data deposition, where you can see that a piece of data can actually be related to quite a number of material entities. And then there's still an organism that has just been existing in time and space, regardless of whether we're observing it or not. So one of the things in the question around what you would do if you had, say, for instance, a living specimen or something, wildlife that you're going back and resampling, or an arboretum, for instance, you can just use our 10 steps, and you can stick in a prefacing step where there's time 0, where the organism is still alive, and then you can model the times at which you come and subsample it, perhaps yearly, year 1, or at different times. So in that way, you can link them all back to this original starting organism. So the way this actually manifests on the platform that we're building as part of the ARGA project is that we can see at the collection event, you'll have a field identifier, and then the accession event will have a different number, and it will also have a different number there when it's subsampled. In this view here, we can see that we can also then model this many relationship here where there were a number of amplification events for this same individual specimen, and we can see that. And then on the UI, people can, oh, I've gone the wrong way, hang on. There, on the UI, people can flick through the different events and get the different metadata around those individual sequence assembly events. What was exciting for us as a project when we started doing this was that we realized that this sort of approach could be then used and in a new context, which was to display different types of taxonomies. So we could extend the idea of having multiples to it by being able to display then different types of classifications that may exist. So the way we could also then use that also to display taxonomic history, so we could show different events that had occurred to a name. So it wasn't just organisms anymore, we could also apply the same thing to names. I'm not going. Oh, I wasn't going to go into that in too much detail because I wanted to give a shout out to the talk that Goran Sturgeoff will be giving later on, and he's done a lot of work around this space, so he can talk about the architecture and how we actually managed to do that. And finally, I suppose I just want to say thank you to the team. The ARGA project team has got some data engineers, but I also wanted to thank all of the people who'd been involved in working with the project over the years to try and get us to the point where we are now. Thank you.
We have a couple minutes for questions if anybody has a question for Kathryn.
Talk more quickly than I thought I would.
Oh, there she is.
Hello.
Oh my God. No questions?
I'm not that shocking, surely.
All right, well, thank you, Kathryn. Let's move on to our next presentation. Oops. Yi-Ming Gan, are you here? Yes, come on up. She will be talking to us about what matters for an occurrence ID and what is an occurrence ID that matters.
Thank you, Stan. Good morning, everyone. Thank you for joining me. So there are many discussions about occurrence ID. It makes me nervous when I feel like I have to talk about meaningful identifiers. So there are only a handful of people who ask me why do I use meaningful occurrence ID? And there are only a handful of them who sat down with me and look at the problem. And I would like to take, my co-authors and I think that there's a gap in the conversation. And that's why we are here to talk about the why. So why does the stability of occurrence ID matter? So from this article, based on my understanding, the stability of GBIF ID depends on stability of occurrence ID. If occurrence ID change, GBIF ID change. And the downstream application that are built based on the GBIF ID will not be able to resolve to the occurrence anymore. So what happened was the data publisher needs to provide a list of mapping of new occurrence ID to old occurrence ID so that GBIF Secretariat could do this mapping and preserve the GBIF ID. Now, there are many discussions about the types of occurrence ID. Should it be meaningful, meaningless, UID, or DOI? But we felt that something is missing. That's the process of occurrence creation, because occurrence is not just a column. Occurrence ID is not just a column in the occurrence table. So first of all, what is an occurrence? So it is an existence of living co-organism at a particular place, at a particular time. So we can already represent the occurrence concept with 4 Darwin core classes. So I want to ask the question, how is occurrence represented in the original data tables? Because we data managers work with scientists and their raw data. So this is the original data that I got for about a survey. So the video is attached to a submersible that performed dive at different sites. So we have the event table, which has the date, the location, the coordinates, and the depth. And then we also have a taxon table. These scientists particularly look for vulnerable marine ecosystem indicator taxa. So she has a list of taxa of interest. So, and then she marked detect, if the taxon is detected in the dive, there will be a tick, otherwise it's empty. So each of the cell is actually a row in occurrence table. Second example here is an annual census data, which means this data set maintained by my data provider will be updated every single year. So the scientists go to some sites in Antarctica of different seasons and conducted some census to count the seals. So these are the species of seals. Again, we see this type of metrics. The count is the recorded count based on the sex and life stage combination. Again, each cell is a row in the occurrence table. A third example is the typical material gathering type of data, where an Antarctic expedition on the research vessel go to a different station in Antarctica. They stop there at a certain location and deployed the sampling gear at certain depth. And they collect sample. Here is sediment sample. And within the sample, they look for certain type of organisms, and they have the count of those organisms. And then they don't treat the information as occurrences, but rather they track the samples. They are put into different jars and transport differently to different people. And sometimes we also got subsampling. For example, among a number of 5 or a number of brittle stars, they take one organism. And then from there, they also dissect the organism into different parts of the body for different examination. So in the previous example, there was no occurrence table. So what about occurrence ID? When is occurrence ID created, right? So the original tables need to be transformed into Darwin core tables because so that it can be published to GBIF or OBIS. And OBIS. And occurrence and occurrence ID only be created in the second step. Now, I do want to emphasize on the left side of this table, it comes with various level of resources and skill sets. Some data provider has the coding skills, some not. Some are just master students who lack the guidance from their supervisor to manage their data. And for some OBIS nodes, there is only one person who does all this job. And a lot of the data sets that I receive are in Microsoft Excel table, but we also sometimes receive tables plus unstructured data in Microsoft Word and PDF document. Well, then what is the role of occurrence ID? So from the downstream application and users up to the Darwin Core Archive, occurrence ID is used to identify a row. But if you want to correct something, we need to correct it here at the original tables because they are maintained by our data provider. So the occurrence ID, so this process here it's done by human, and occurrence ID serves to identify a cell or a row in the original tables. That's why I use meaningful identifiers here, because it helps me to go to this cell. And then I know, oh, this is the event that they're talking about. And oh, this is the taxon that they're talking about. And for this example, the occurrence ID is a combination of event ID, location, species, age class, and sex to identify the cell. So will meaningless occurrence ID be able to achieve the same goal? Maybe. And what happens, and I also wanted to talk about what happens to occurrence ID upon dataset update. So again, update happens here because these are the tables maintained by our data providers. But occurrence ID only created here in the second step. If there is no some tracking mechanism in place, an occurrence ID that is created independently of the original table, for example, if we just put UUID as occurrence ID at this stage, then every update will actually regenerate UUID for all of the records. So I would like to end my presentation with these two questions. Should the stability of DRUBIF ID be data context driven? Or should it be independent of content and the responsibility of the data provider who does not maintain an occurrence table? Thank you.
Do we have time for questions, if anybody has any?
Sorry, if someone else wants to go because I've spoken before, no? Happy to give my slot. This is great stuff, and I would like to repeat what Derek Sykes, many of you know, and databasing entomologist from Fairbanks, said in the International Entomology Conference in 2016. He was basically talking to entomologists who don't database to exactly very same topic, persistent identifiers and occurrence IDs, and his argument, which I like very much, was that IDs should be generated before you generate data. There are empty rows in the data system, CMS or whatever, which are predefined and you're not allowed to play with. The same way when you apply for the passport or you receive a social security number from your municipality, this is not the number you play with, you just get a number. And I just came from the International Entomology Congress in Kyoto. There are 4,000 people, of which maybe 1% will be able to database at the level that you people database. And this number will not change very rapidly. So there must be a kind of entomologist proof or biologist proof system that generates occurrence ID, like you shown, very early. And so this is like an old thought, a purist thought that we should not play with occurrence IDs. And I think they should not be context specific. They should be given by an authority that we don't have. They should be minted externally before you generate the data. But that's very hard to achieve because it's very expensive. And maybe now you want to say something, Marcus, I can see. Yes.
Just from the same organization, Marcus DBIF, I'm just the exact opposite thinking, because I think it's great that you have a compound key, basically. You have tons of tables that you have to combine to create that row that you don't track specifically. And I think it's perfectly fine what you're doing there to use this compound key. If it's content, if it's not opaque or not, I don't really, I mean, it's IDs that you combine. It's just a compound key in a database. And that's, to me, perfect. Yeah.
Thank you.
I was going to come in the middle of that and say, if an ID is a primary key, then should it be updatable? So if you do change the data for the compound key, are you creating a new occurrence in that instance? So I'm sort of in the middle. It doesn't matter as long as the key is immutable.
Thank you. I don't really understand that. Yeah, thank you. Yeah, all right. I hope the message was clear. Thank you.
Our next speaker is Ben Collier, developing a complex object model, seeking a coherent, integration-ready collections management data model, a data architecture story.
punchy. So morning, I'm, as Ben Collier, Senior Data Architect on the RECODE programme at the Natural History Museum in London. And I'm going to talk about challenges we face developing a CMS model. So while it's not directly related to the concept of organism IDs, I think some of the things we explored are related in a general way, and we will touch on that subject as well. So this was a big, complex challenge. So we're designing a new CMS for the Natural History Museum for the whole collection of 80 million specimens. We have lots of user requirements. We've generated nearly 1,000 user stories with many functional requirements that lead to data requirements. And we have to create it with a good user experience for the CMS users. And we are constrained by, we have an existing data set that needs to be, which is digitized, and there are data that are yet to be digitized. And the process of dealing with that existing data is my colleague's Marla Spencer's talk in Contributorial 1. So if you're interested in more of that detail, please go to that. And yes, so again, this will be a preview of what the data model is like, just an introduction to it. So this is Steen DuPont next door. This is an overview of the RECODE program that is happening right now over there. And this is a whole program, and we will be focusing on the data model section, data modeling here, but the backbone applies to all areas. So it includes this work. So our main role as data architects is to develop a logical model to present to our software partner to implement in their product. But obviously the work of the data modeling area does overlap with adjacent areas in this diagram. So what is the role of the CMS in the context of this joint conference? So curators want to curate specimens, count them individually and describe them fully. Aggregators want to share data for multiple specimens and find commonality and track changes, among other things. There are lots of different ways to categorize and describe specimens in human readable format, but when we share data, it's largely done by machines running defined, scheduled processes. So our feeling is the CMS should be designed to act as a bridge between how humans interact with data, with complex descriptions, and sharing that data between machines in a consistent manner. So then we talk, what is a specimen? Now, the word specimen has many different meanings in the curation community. So I only use it in context of describing things. It's not really used as a term in the model because it means lots of different things. So we consider what can be handled, what is loanable and has a specific location in the collection, and we call this a collection object. Then we consider what can be, what can be classified taxonomically, and we call that an identifiable item. And finally, we consider relationships between these two concepts and between themselves. These relationships can be complex, but we also need to ensure we represent the simple simply, because lots of specimens will just have a simple relationship between these, and we don't want to overcomplicate the system. And we need to define, there's a chronology of how things happen, but it's also represented as a circle. So a biological geological individual is observed and recorded in its natural habitat. It is collected by a collector at a specific place and a specific time. It's then added to the collection, and in our terms of one or more collection objects. And then each individual instance of a species within it, or those objects, is determined to be of at least one taxon name. Opinions vary. but the individual of a species is what occurred in nature. But it's not necessarily all of what was collected as a unit. It could be a leaf with a gall, a rock with moss on, or just part of an individual, for instance, a bone of a fossilized animal. So this is the complex object model we have designed. So we have the collection object, which we define as the physical object or part of it that has or had a specific location in the collection. We have the identifiable item, the biological, geological individual of a single taxon that is represented in part of one or multiple collection objects. And these are linked by what we define as a composition record, as the details of how an individual is represented in a specimen or specimens, or how a specimen is comprised of individuals. And this is a resolving class because the relationship between object and identifiable item is a many-to-many relationship. And we can relate objects to each other as collection-object relationships, as a link between a single source object and a single target object, and the details of any physical actions were undertaken in the process. So this is where a process that creates the object is defined. So preparation defines a process rather than an outcome, which is just another collection object defined through this self-join. This work does share similarities with work James Macklin did many years ago at Harvard Herbarium, and there's lots of similarities in the model, so I'd just like to credit him for some of the useful work he did there. It's been very helpful. And then we come into how do we define and describe a collection object. We can use lots of types of objects, so we use subtyping. There are many different ways of doing this. Different types of objects share many attributes, but we need to be clear on the differences. Some attributes may be vitally important in one area and irrelevant in others, so we have to be clear on those descriptions. And there are sometimes simple hierarchies of descriptors, but sometimes multiple overlapping ones with complex conditional logic. We need to describe what things are rather than how they are governed in the collection. So a slide is a slide is a slide. It doesn't matter what's on that slide. Its object descriptor is a slide whether it has a thin section of rock on it or a plant sample or multiple other diatoms on it. And object can be transferred between departments. So this shouldn't be delayed by data constraints imposed in one area that aren't relevant in another. And of course, it doesn't mean that origin is unimportant. The way objects are curated in different areas are very different. We do things to botany samples that petrologists are not interested in. So we do have to understand there are those differences. And then we come to the point of what is an individual. So earlier, I defined the identifiable item as the biological or geological individual of a single taxon that is represented in part of one or more collection objects. But what is an individual? An elephant is easy, we can give them a name. But in this photo, what is one grass, one rock? So in our model, we make a practical rather than philosophical definition. Two objects can share an identifiable item if a determination about one must imply the same determination about the other. This did lead to other complexities about how we uniquely identify the identifiable items, where we have something like a complex object, like a slab of fossil matrix with multiple individual fossils in it, that need to be uniquely referenced for publication about those individuals. As a project, we have developed many different use cases, a dozen generalized scenarios based on hundreds of stakeholder requirements, and defined how the data model can accommodate them. Within the constraints of the model, there's still multiple options to describe and link objects. Each one of these scenarios is used to test the model and inform the UI how to link entities to create business objects that are presented to the user. Everything is linked. The data model has many paths how to link entities, all the entities across the model to one another. So there needs to be controls about how this is done in the UI and other interfaces. We need to find relationships in the model that are required to keep the data consistent. The combinations of relationships in the model can be interpreted as single descriptive relationships. For example, we don't need to define a relationship shares collecting event between two objects if they're both linked to a parent collecting event. We're creating a normalized data model to allow for maximum flexibility and underpinned by a robust single version of the truth, or truths, because opinion varies. So in summary, we aren't trying to solve all the problems in the data model, but to enable solutions. It's not up to the data model to redefine standards, so where they vary even across an institution, the data model must be able to accommodate those. And there is a balance between the data model solving all problems and enabling wider solutions. There's a governance layer on top of the data model that guides users or processes how to enter data and read data consistently. We need controls on the data based on rules and regulations as well as data consistency. We need to know the difference between logical, context-specific constraints and physical constraints. And overall, we need users to enjoy using the CMS. That doesn't mean giving them what they want, but rather what they didn't know they needed. And in constraining what they can do in a well-designed way should lead to a better user experience and higher quality data for the curator institutions and the natural history community as a whole. Included in this is controlled values list, so I'd like to direct you to another talk that Jutta Buschmann's giving in SYM13. There are more details on that. And we have to understand that the data model is never finished. It must be adaptable to change and under constant development. So I'd thank you for your time, and I have plenty of time in the conference for further detailed questions and lots more information.
And we are running well ahead of schedule, so plenty of time for questions.
Thanks very much. Really interesting talk. Elspeth Haston from the Royal Botanic Garden, Edinburgh. I'm really interested to see how you're developing the CMS. I guess one question is just I'm interested to know what you're actually connecting in terms of Darwin Core, and where the connections are.
So I see a Darwin Core record as effectively a business object on top of the data model. So we will define paths for mapping the data model to the whole set of Darwin Core records for data sharing and other standards where applicable. That's still under development, so we have it in our existing CMS, so we see the rules there. But I see from these talks that a lot of the concepts fit in quite nicely. So my feeling is we are able to do it, and I have been referencing Darwin Core throughout the process.
Super. Thank you.
Yeah, well, it's an ancient spinach question. I wonder what's your solution for the mixed samples. Lichens on the rock, you never collect one species at a time. There are 7. And modular organism, I love your example with the elephant, but I work with fungi. There is no such thing as an individual, only genette or ramet, whatever. So like this kind of things, I guess it's dangerous to say things like this, but specimenocentrism is a problem to generate a universal data model. And like people who don't work with elephants, like me, would like to know kind of how far your schema can reach and where's the limit of applicability?
That's a very, very good question, something we should be considering in our modelling scenarios about how we deal with that and how you assign a taxon name to a mycological specimen, and where we define that, and if you split that into separate objects, do you want to share those taxonomic attributes across those for ease of use, so keep that truth, or do you? I I can't say this, but is there variation across a sample? If funguses change it, where do you do that? I've used examples I've seen in mineralogy. You have the same idea where type specimens can be tiny little points on the rock sample, so it does get very complicated, and that's where I say it's a practical decision about how the individual want to record that in the CMS. So it does need exploration, but you are right, that is at the limits of where the model is now, and it's going to be fun exploring that.
Well, faced with the decision to hold up and give ourselves 5 minutes, we should be starting the next talk in 5 minutes. And I was told not to let ourselves get too much ahead because people are trying to jump. So, talk amongst yourselves. Bio break. 5 minutes.
I don't know whether people online got your message. It's tricky.
Hi, James. I don't know if you can hear me. You're up next, but
I can hear you, Stan, but just play my recording.
yeah, we'll do that. I'm just letting you know that we're told not to get too far ahead of ourselves, so we're going to just have a 5-minute break.
Yeah, that's fine. I think that's a good idea.
All right, thanks.
Enjoying the show so far. Hello to all.
All right, we're going to get started again, but before we do and before I let you go, I want to draw your attention to when you see this, the program, if you view the program for Oxford Abstracts, up here in this corner is a little bubble with dots. This is the chat. There is a group chat for every session. So our session here is SYM25, SYM25. You can search for that in a group chat. Would all the speakers please join that group? And then you will be able to see if anybody asks you a question. Okay, next up is a recording from James Macklin, but he's online, as you heard. And he will be, where his talk is entitled Challenges, Case Studies, Candidate Solutions, and a Wish List for Interoperability of Occurrences through a Modern Collections Management System. Go ahead.
I hope you've enjoyed the talk so far in our symposium. My name is James Macklin. I'm Agriculture and Agri-Food Canada colleagues David Shorthouse, Christian Jean Dro, and Satpal Bilku will present some challenges, case studies, candidate solutions, and a wish list for interoperability of occurrences through a modern sample management system. As has already been described by the earlier speakers, a Darwin core occurrence is an existence of an organism at a particular place and time, thus requiring a Darwin core organism and event. The main driver for this symposium and talk is based on the many discussions my colleagues and I have had about the nature of occurrences and how to repeatedly generate them with stability in our new sample management system, DINA, that we have been building in partnership with the consortium. Our novel simplistic data model is strongly influenced by the material sample, now material entity, as a core object. This allows for considerable flexibility in the kinds of objects that can be managed. Here, the parent sample of beetles in a jar is subsampled to investigate the white beetle, which undergoes several more subsampling downstream through various processes, including a morphological determination, a sequencing process, and a molecular determination. These are all tracked with a robust provenance scheme, which maintains our relationships back to the parent and the original sampling event and its event metadata. Our challenge is to produce an occurrence based on this model, or perhaps not. We see two challenges with the Darwin core occurrence in a simplistic data model. First, Darwin core organism implicitly requires Darwin core identification, but only some material entities within a chain of subsamples are taxonomically homogeneous. We've coined this the shadowy creatures. Second, Darwin core event metadata are subject to refinement and reassociations with material entities. We've coined this event drift. This is a snapshot from the DINA user interface with some annotations illustrating how a collecting event with a sample of organisms using this trap is later subsampled for a particular organism of interest and a determination made. This organism, the fly, maintains its relationship with the parent and the associated event metadata. However, when you give data curators the freedom to fix an error in what data are present on a determination, non-syntactic or non-semantic, then the scientific name that was once shared could be altered for the next data publication event. The concept of the Darwin core occurrence will have changed, but the alteration may be an illusion due solely to minor improvements to data quality. This example is parallel to the first one, with the exception that this involves living organisms, and more than one organism is being tracked that is associated with a single material sample record. And this example illustrates a material sample that has a morphologically identified organism and also, through a subsample and sequencing process, has a second determination that may or may not agree with the morphological one. How do we represent the two organisms with conflicting identifications where choosing either one eliminates valuable information? For biomonitoring research, it's typically the case that a living material entity or an environmental sampling of soil or water is done over a span of time. For example, a living material entity, example a tree in a nursery, is examined for nematodes over time, as is depicted in this picture. Multiple time series of material entity tissues, each with Darwin core event and implied Darwin core organism, the host, with Darwin core identification. Each material entity, host tissue per time series, with multiple material entity, subsampled from tissue, each with Darwin core organism having Darwin core identification, the nematode, and or indicated as a Darwin core occurrence status equals absent. Environmental DNA samples can be split into many subsamples containing one to many organisms, which may or may not be identified, and identifications may not be associated with known organisms, leading to the use of folksonomy labels. Drift in collecting events post-publication can happen in many ways. Material samples may be added to existing events or removed and reassociated with another. Existing event terms like date, collector, or aspects of place can be later curated to improve precision, fix errors, and other quality-based issues. The new proposed GBIF data model allows for better relationship management across entities, but maintains the concept of occurrence and its relationship to an event and an organism. The question here is, what might GBIF do if a data provider does not have an occurrence ID? Will this continue to be a requirement to publish? So we see some constraints on expectations for data sharing. First, the Darwin Core occurrence ID, despite a plea for global uniqueness and stability in a publicly shared data set, is inherently unstable locally because of event drift and those shadowy creatures. A meta-concept has no operational utility in the local management of samples. Business logic to repeatedly construct these ephemeral data objects is expensive to develop and maintain, and it's a barrier to communicate data enhancements with stakeholders. Here we suggest some candidate and potentially provocative solutions for aggregators. Darwin Core Event ID and Darwin Core Material Entity ID are locally stable, do have operation utility, and are user-facing in data curation interfaces. So Darwin Core Occurrence ID should be made optional in a Darwin Core archive and upcoming frictionless data exchange standards. We should leave it to aggregators like GBIF to construct Darwin Core Occurrence ID from constituent Darwin Core event and Darwin Core organism based on its business logic for its end users, not that of the data providers. And communications with data providers, i.e. round-tripping enhancements, should not refer to Darwin Core Occurrence ID when none was provided. This is like having a sample-based data producers construct Darwin Core Occurrence from GBIF would be functionally equivalent to asking that they maintain work affiliations for all known people in their system if they could publish to ORCID. It's impractical and errors do propagate. So we have a couple wishes to promote discussion. That sample management systems like DINA stay the course without Darwin Core Occurrence because they can remain flexible to the needs of research. Also, the clarity from GBIF and other aggregators that index and serve valuable sample-based biodiversity data, which include specimens, i.e. whose responsible is it to create a Darwin Core occurrence? Finally, we'd like to acknowledge our partners in the DINA Consortium, the AFC DINA development team, and our science and IT colleagues and management for their support. Thanks very much.
So are you with us, James?
In what sense, Dan?
That we can hear you, you can hear us.
I guess that is true.
It is true, yes, we're all good. Does anybody have any questions for James? We have a few minutes. Everybody was quite pleased with your talk, very happy.
Hey, that's great.
All right, well, again, please look for comments in the chat in Symposium 25 and have a nice coffee break and a lovely rest of the day. Thank you.
Thanks, Dan.
I'll stay here. So